the extreme bottom right is Edison. Edison's work was driven by the idea of illuminating the surroundings. He drove with single-minded devotion towards illumination, but in the course of his research, he did define the electron. It is there in his book. It's called the Edison effect, but he did not pursue work towards the electron, but instead he pursued his work towards the light bulb. J.J. Thomson discovered the electron and won the Nobel Prize. Edison was an inventor. Bohr was a fundamental scientist. But what was Pasteur? Pasteur now was both a fundamental scientist and also an extremely applied scientist. Pasteur's work on the foundations of modern organic chemistry, stereochemistry, the relationship between three-dimensional structures of molecules and optical activity or chirality are in fact fundamental to the growth of organic and biochemistry. On the other hand, Pasteur is also the founding father of microbiology. Without Pasteur, our ideas of infectious disease would not be what it is today. In fact, we have honored Pasteur by calling the process of sterilization of milk and all as pasteurization. Therefore, that is the goal towards which everybody should strive, and that is what Donald Stokes said. In his book, in his unpublished manuscript, which was published later, this is a black and white figure without all these pictures and all. I have simply colored them up with Google Images to make them more attractive. But I was asked myself the question, what should I put in the bottom left quadrant, which Stokes left unnamed? That is what average academic and industrial R&D lies. Vilas Shankar is here. He led a R&D laboratory in Bangalore many years ago. I think most industrial R&D labs at best could be called average. One would certainly not put them into the same category as the laboratories of Bayer when the ammonia synthesis was done, or the Bell Telephone laboratories when the transistor revolution took place. What image could I use? I can only find Lakshman's common man. And therefore, now it turns out that our institutions must move in this academic space to find their way elsewhere. They can go now forward. So on the next slide, I would ask, how do you extend the Stokes diagram? Now, if I ask my colleagues at the Indian Institute of Science, so not to be accused of being parochial, I have put my own building where I spent a decade right there at the bottom. And if I ask myself the question, how do I move this? And I'm not showing you this picture after retirement. I showed this picture in 2008, and the Indian Express picked it up, and I was later on asked in a Senate meeting at the Institute, did you really say that the Indian Institute of Science was in the bottom left quadrant of this thing? I said, yes, I did. It is there, and it has to move. And, this is the kind of thing that the government even today will be saying. But the government believes that if you give 1,000 crores over 5 years, which is not a great deal of money, that institutions will move up. That's not what happens. You still need to create what your symposium has put so nicely in the title, so eloquently in the title, create a conducive environment. And I think your task really is to find out how do you create this conducive environment. If I ask my colleagues, they will say, you must have unlimited administrative flexibility. That is, every faculty member should be allowed to do whatever they like, and you must have unlimited financial resources. Now, both these, any administrator will know, is an impossibility. So then one might ask, what can I counter them with? I will counter them by simply changing the axes. I will say there must be teaching and there must be research, and if you have maximum teaching and maximum research, you will have an ideal university. That is what the great universities of the world are today. Where there's a great deal of wonderful teaching and there's a great deal of wonderful research. Many times not done by the same person, but sometimes, occasionally, done by the same person. Many of our institutions do not teach, and many of our institutions do not do research, and sometimes when they do both, they do it badly and remain in the bottom left quadrant. Mahendralal Sarkar, the man who first thought of creating a research institution in India, before Jamshed Tata, created the Indian Association for Cultivation of Science, which sparkled briefly when Raman was there, but afterwards, once again, the flame was extinguished. He said, the world's chief and greatest discoveries have been made by those who have had or have to teach. Educational institutions still furnish the best laboratories for research. Pure research institutions have only recently come into existence. This is very important, even if one is going to talk about eventually creating a new institution for Ayurveda. Because my worry is that, for example, with the kind of environment that we have in India, Ayurveda now is in fact a politically correct subject to study. And when a subject is politically correct to study, it will turn out that government can in fact allot large sums of money to create a new institution. It might be a very worthwhile subject to study. But when a large sum of money is allotted, you must now create an institution which is worth that money, which eventually justifies its existence. And therefore, it must understand the parameters under which it is in fact created at the beginning. Today in academic space, this is the problem. I can again renumber the axes. The Stokes quadrants are in fact can be used again and again. You can have quality of governance and academic performance. The institutions with the highest academic performance, also with the highest quality of governance, are the ones which are going to end up in Pasteur's quadrant. If governance is poor, academic performance will almost always be very poor, except for isolated pockets of individuals who work there, but that doesn't make any difference. But I will come back to Ayurveda for a minute, because I did do a little bit of homework. And when I did a little bit of homework to understand what AYUSH really was, I must confess that I never really expanded the abbreviation AYUSH. And I might be prejudiced in this, but I never did. But then when I went to the ministry site, I found all of this. I now color-coded them in such a way that I have changed the colors here. They use Ayurveda at one extreme of what might call traditional medicine. And there is homeopathy at the other end of what might be called traditional medicine. I think it is, in fact, in many ways, the entire spectrum of the AYUSH disciplines is incompatible with the tenets of modern science. Now, I would certainly not. Now, in no way is Indian tradition involved with homeopathy. And this is something that one must understand, because homeopathy itself is a German invention. And if it is a German invention, we should have left it to the Germans to actually promote their traditional practices. And we should not have worried too much about it. Ayurveda, on the other hand, is traditional Indian medicine, and therefore it deserves every effort at promoting, investigating its causes and its effects. But I found there was this interesting controversy which now goes on about the name AYUSH itself. And levers, which now is well represented over here, actually is now involved in a legal battle with the government of India on AYUSH. And this might be a very good opportunity to leave the name AYUSH to levers and change the name of the ministry itself to reflect more accurately what it is supposed to be doing. But eventually it turns out that there are natural products. Today, for example, levers in competition with Patanjali now puts advertisements like this, where everything in nature is considered to be good, and everything that comes out of a synthetic chemist's laboratory is considered to be bad. And as a chemist, I find this extraordinarily surprising. Curcumin, of course, now is advanced as everything. I want to ask the question here, are all naturally derived products Ayurvedic? And there is, I think, some kind of misleading advertisements now which are all over the place and with which the common public is in fact fooled. In my post-retirement phase, I have been going around talking about chemical myths in advertising which we must resist. And chemistry is unavoidable. All these molecules are natural, and all these molecules have uses. And sometimes I found that the Colgate toothpaste is supposed to have all these constituents. And all this tells you is that chemistry is sort of unavoidable in this. Whether they have them in the right proportions or not, I do not know. But plants do not produce molecules for your benefit and mine. And this is where I think human beings must be considered in the larger context of biology. We have a Minister for State for Human Resource Development who actually denies any kind of natural selection as the basis for our biological present. And it turns out that we can learn a great deal about evolution by studying plants, animals, bacteria, all living creatures. Now, are the alkaloids which are here, are they beneficial? I don't believe that any of these you would consider today as beneficial. Some of them you would consider as positively harmful, but all of them are produced by plants. There are a huge number of other chemicals which are produced by plants, and many of them in mixtures and in very special plants are in fact the basis of many of the therapies of traditional medicine. Paracelsus, probably the man who began much of modern chemistry, said this well. And this, I'm sure, is written in Sanskrit, which I do not know. In Sanskrit, I'm sure you will find many, many such statements. He says that everything is poison, there is poison in everything, only dose makes a thing, not a poison. And this, I think, is the basis of formulations which many of you will study. Between natural and man-made chemicals, there is only this difference, toxicity. But toxicity depends on the dose at which you take something, and therefore there are many natural chemicals and natural products which are as toxic as synthetic chemicals and synthetic products. But look at two of the most common things in India, the red chili and turmeric. They have molecules which look remarkably similar, and today modern science tells us that this part of the molecule is recognized by a class of membrane receptors called the TRP ion channels, transient receptor potential ion channels, and therefore even the physiological basis for their action might today be understood. So there would be a clear connection between cell biology as it is practiced today and Ayurveda and Ayurvedic practice as it is evolving in the modern era. I'll tell you only one example, and this I think many of you might have come across, but I will tell it in my own words. That is UU2's discovery in China of artemisinin as the anti-malarial. The time when the Chinese were trying to find artemisinin, they were trying to find an anti-malarial by screening plant extracts. They screened plant extracts because the Chinese army was planning to face the Americans in Vietnam, and more Chinese would have died of malaria than of American bombs because there was thick forest cover in the jungles of Indochina. They had the desire to find an anti-malarial. They took thousands of plant extracts and injected them into malaria-infected mice and looked at parasite clearance. This one plant extract worked, but it did not work reproducibly. The results were irreproducible. Sometimes it worked, sometimes it did not. It is the cause of the irreproducibility that UU2 investigated. What she then did is she went back to the ancient Chinese literature and translated from the Chinese and looked at the way in which the plant was being used in traditional medicine. If you give an organic chemist, and I was trained in organic chemistry, a plant extract, what I would do as default is to put it into a flask, pour some organic solvent on it and boil it. This is why something is always boiling in a chemistry lab. And I will make a decoction out of it the same way you make a decoction out of coffee or tea. But it turned out that the old Chinese literature simply said that you put the plant material in water, squeeze it, and then drink the decoction. So it was a decoction made out of a cold water extract. So all that was done here now in making the extracts reproducible was to stop the process of heating. And then they discovered the molecule artemisinin. The extract was a crude extract which was administered. The structure of artemisinin came many years later, and the structure of artemisinin is the molecule that I've drawn at the right. But when you look at this today, this is what the organic chemist would call a terribly complicated structure with an organic peroxide. But peroxides are substances which even with elementary training, any chemist will tell you is explosive. Hydrogen peroxide and other kinds of peroxides are what the Indian space program uses in their rocket propellants. So peroxides have the ability to explode, produce hydrogen and oxygen, which can then combine and give you combustion. That's what was happening to this molecule when heated to a decoction. It was losing its activity. So here you can see a merger now of modern science and ancient literature, but properly translated, properly read, and properly interpreted. I'm coming to the end, and here all I'd like to say is, the only things that I know about Ayurveda are the times when I have induced Professor Valyathan to come and speak, or asked Professor Valyathan to write an article for Current Science. For many years, he was also the president of the Current Science Association, so he would come annually for a meeting, formally to approve the accounts of the journal. And the rest of the time, he would say something and talk, and it was always a great pleasure and a great learning to listen to him. And in an editorial that he wrote in 2006, he actually said that Ayurveda means science of life of which medicine is no more than a part. He also said, no wonder P.C. Rai called the interval from 600 BC to 800 AD the Ayurvedic period, because Ayurveda was the cradle of not only medicine, but also of chemistry and sciences of plants and animals in India. My reading of this now, I went and gave a talk for P.C. Rai's 150th anniversary at Kolkata University and unveiled a bust. I must say I had not read this at that time, but really in many ways, P.C. Rai's book was the way in which chemistry, botany and zoology really saw a common connection. And today, if you have a Center for Interdisciplinary Health Sciences, this is the kind of integration that one must really aim for. He said a lot of other things, but he also says that it's not only for finding molecules, for finding extracts, for purifying them. It's not only that kind of pharmacology which is important. He says, for example, dosha prakritis are specific and determine the manifestations of a disease and the response to treatment in individuals. Do these prakritis or phenotypes have genomic counterparts? He asks this question in this editorial written in 2006. And it's a great coincidence that after I got the invitation and I was thinking about what I should say here, this issue of Science, which is the 10th August 2018 issue of Science, one of the leading magazines of modern science, had this article, The Future of Humans as Model Organisms. And the article begins with a quote from the Nobel laureate Sidney Brenner. And what Sidney Brenner said is, we don't have to search for a model organism anymore, because we are the model organisms. And it says here, a collection of human phenotypic data and its integration with omic information, genomic, proteomic, all the other omics, along with remote sensing data, could provide extraordinary opportunities for discovery. A comprehensive human phenomic science approach could catalyze this effort through both large-scale light phenotyping studies and deep phenotyping studies performed in smaller groups of individuals. I think it is this paragraph that practitioners of Ayurveda, the specialists in Ayurveda, really have to study and ask, what are the connections between modern science at its very frontiers of biology and what Ayurveda for a long time has espoused as the importance of looking at the phenotype of a patient before actually prescribing a course of treatment. Lastly, I must show you this slide because I was trained in chemistry, and my hope still is that any resurgence of Ayurvedic research will also lead to a resurgence of natural products chemistry, which was one of India's strengths in the 1960s and today is a major area of weakness. In fact, I would call it a major area of darkness after V.S. Naipaul, who recently passed away. But it turns out that chemistry is really the lingua franca of the medical and biological sciences, irrespective of whether you are going to study traditional medicine or modern medicine. Traditional medicine is based on empiricism. So was modern medicine. Modern medicine looks for cause. It looks for mechanism. And in modern Ayurvedic research, you are also going to look for mechanism. What Arthur Kornberg, one of the founding fathers of molecular biology, said, analyzed this. He said there was a rift between the cultures of chemistry and biology. And he said that this rift might derive from the fact that the chemists used this side of the brain while the biologists used this side of the brain. The left side of the brain is supposed to be the one responsible for analytic abilities. The physicists and the mathematicians And I suspect the engineers would put themselves very firmly on the left. The biologists, the poets, the literary figures would put them very much on the right. The chemists are somewhere in between. They are schizophrenic most of the time. And now you might ask the question, where do you put modern medicine and where do you put traditional medicine? And how do you really integrate these two sides of the brain in thinking about creating a conducive environment for modern research? But I hope that in thinking about this symposium will eventually result in catalyzing the drive towards a truly modern institution, which integrates the best features of the old, the best features of the new, but also adopts some features of the immune system which have made the Indian Institute of Science resistant to the Indian political environment. Thank you very much.
Thank you, sir, for sharing your excellence experience. Sir, 1 second. About IISc, we specially thank you for providing your 19 selective editorials for our symposium compendium. I would like to request Dr. M.J. Dev, who is the great scientist and mentor of our AYUSH Center of Excellence, to come forward for the felicitation. No, sir, no, sir, please. We have just received a message from Dr. Vijay Bhatkar saying that he has conveyed his apologies for because he could not able to attend this August gathering. He has wished a great success for the symposium. Now I request Dr. Sanjay Dhande to come on dais for the speech. He is receiver of Padma Shri from Government of India. Professor Dhande was director of IIT Kanpur from 2016. He is a chairman of Board of Governance of NIT Delhi and National Institute of Industrial Engineering Mumbai. He is member of Scientific Advisory Council to the Prime Minister of India. He has enormously worked on computers and technology for the betterment of society. I request him to tell us about why IITs stand out.
Well, I must say that after Balram to speak is a tough call, actually. But the topic, namely regarding why IITs are so successful, is a very dear topic to me, and I would like to sort of give my thoughts to you. As you know, the technical education is almost 5 phases in the history. The first phase is the pre-British era, where mostly the Indian engineers were trained for supervisory work, and therefore it was predominantly civil engineering oriented. Thomson College in Roorkee, or the Engineering College in Pune, or the Guindy College in Madras, or the Bengal Engineering College, where few institutions where supervisors or overseers were trained. This went because the dams, the canals, particularly the Ganges Canal, and the railways were the predominant structures for which supervisors were required. Then came the Second World War, which